Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of the year 2012. If you are new to my channel, please do like, share and subscribe. Coming to the year June 2012. First question, these are questions from part B. Site-specific recombination results in precise DNA rearrangements, which is limited to specific sequences. The enzymes that are important to carry out the process are, so what are the enzymes involved in site-specific recombination? So they are nuclease and ligase. Answer is option 2. Then next one, which of the following statement is not true about small interfering RNA? siRNA has a 21 to 25 nucleotide sequence with 2 nucleotides overhanging at the 3' end. That is correct. siRNA is processed by the RNA protein complex RISC. That is not correct, and that is your answer. siRNA is often induced by viruses. That is also correct. siRNA does not generally act at the level of transcription. That is also correct. So they have asked for the wrong statement, and the wrong statement is option 2. Then next one. Presence of an internal ribosome entry site, IRES, in mRNA. It inhibits, so IRES is allow cap independent translation and is usually under adverse conditions. So it inhibits its translation, promotes its post-transcriptional processing, has no impact on its translation, promotes its translation under adverse conditions. That is correct. Option 4 is correct. Regulatory elements for expression of ribosomal RNA genes reside in the, so they reside in the non-transcribed spacer region. Option 2. Then next one. A gene encoding tRNA undergoes a mutational event in its anticodon region that enable it to recognize a mutant nonsense codon and permit completion of translation. Such a mutation is known as, so a gene encoding tRNA undergoes mutational event. What happens due to mutation? Anticodon region that enable it to recognize, and the mutation is in the anticodon region, and because of the mutation it recognizes a nonsense codon and result in completion of translation. So such a mutation is called as nonsense suppressor. Answer is option 4. Then next one. These are part C questions. Double-stranded DNA replicates in a semi-conservative manner, that everybody knows. In an in vitro DNA synthesis reaction, dideoxy-CTP and dideoxy-CMP were individually added in excess. That is, they have taken 2 separate reaction tubes. Into one they added dideoxy-CTP, in another they added dideoxy-CMP, in addition to DNTPs and other necessary reagents. Rate of DNA synthesis was measured by incorporation of H3 thymidine. So they want to see what will be the rate of DNA synthesis. So they incorporated H3 thymidine in the tube. The 4 graphs drawn below represent the rate of DNA synthesis in 2 separate reaction tubes. Which of the following graphs represent the expected data? So CMP is a monophosphate and CTP is a triphosphate. And DNA polymerase, it recognizes a triphosphate form of the nucleotide, not monophosphate. Okay. So when DDCTP is added, it results in chain termination. Just like your Sanger sequencing method, it results in chain termination. Whereas when DDCMP is added, there won't be any chain termination because it recognizes only a triphosphate, and you have added normal DNTPs also along with CMP. Dideoxy CMP, you have added normal DNTP. So CMP will not be recognized by the DNA polymerase, but the normal DCTP will be there. That will be recognized and that will carry out the process of replication. So here rate of DNA synthesis versus time graph is given. These triangles, they represent rhombus shapes represent DDCMP and circles represent DDCTP. Okay, so the first answer is option 1. Why? Because I told you already that there won't be synthesis of DNA synthesis when dideoxy CTP is added. So the first graph will be for CTP will be going down like this. It will start, and then once DCTP is added, it won't be, there will be chain termination. So this will be the DCTP graph, and in DDCMP it won't be recognized by DNA polymerase, but other DNTPs are present, so they will be incorporated and replication will continue. So this will be the DCMP graph. So the answer is option 1. Then next one, the question is very big, but what they have exactly asked is only how Okazaki fragments need to be matured into continuous DNA strands by which combination of enzymes. So we will read the question first. In semi-conservative mode of DNA replication, two parental strands unwind and are used for synthesis of new strands following the rule of complementary base pairing. That's correct. Everybody knows about that. It is semi-conservative mode of DNA replication. Synthesis of complementary strand requires that DNA synthesis proceeds in opposite direction, while the double helix is progressively unwinding and replicating in only one direction. One of the DNA strand is continually synthesized in the same direction as the advancing replication fork and is called leading strand, whereas the other strand is synthesized discontinuously in segments and is referred to as lagging strand. These short fragments made discontinuously are labeled as Okazaki fragments. These Okazaki fragments need to be matured into continuous DNA strands by which of the following combination of enzymes? So your question is only one sentence, that is Okazaki fragments need to be matured into continuous DNA strands by which of the following combination of enzymes? So you need DNA polymerase 1 and DNA ligase. DNA polymerase 1 is for primer removal and to fill the gap. Primer removal, and DNA ligase is for filling the gap. So the answer is option 2. Then next one. A synthetically prepared mRNA contains repetitive AU sequences. That is, it has AU, 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 AU like that. The mRNA was incubated with mammalian cell extract which contains ribosomes, tRNAs and all the factors required for protein synthesis. So this mRNA which is having this synthetically prepared mRNA which is having repetitive AU sequences was incubated with a cell extract containing ribosome, tRNA and all the other factors that are required for protein synthesis. Assuming no initiation codon is required for protein synthesis, which of the following peptides will be most likely be synthesized? So you can get AUA, UAU, AUA, UAU. So two peptides, two amino acids will be synthesized. So a single peptide with alternating sequence of two amino acids. That is the answer. Answer is option 2. So coming to the next question. The lac operon in E. coli is controlled by both the lac repressor and the catabolite activation protein CAP. In an in vivo experiment with the lac operon, the following observations were made. CAP levels are high. Repressor is bound with allolactose. CAP is interacting with RNA polymerase. Which one of the following conclusion is most appropriate based on the above observations? So CAP levels are high means your glucose is absent. Repressor is bound to allolactose means there is lactose present in the medium, and CAP is interacting with the RNA polymerase. So the answer here is glucose is absent and lactose is present. Answer is option 4. Then coming to the year December 2012. These are part B questions. Coming to the first question. With reference to lac operon, what will be the phenotype of an E. coli strain having the genotype I minus O plus Z plus Y minus I plus OC Z minus Y plus? So if you see here, this is I minus, whereas this is I plus. And I, you know, it is the repressor element, and it will code for the repressor, and it is transacting also. So I plus is over here. So this I plus, it will produce the repressor which will bind to this operator. It cannot bind to this operator because it is mutated. It is constitutive operator, OC. But this is normal operator, so it will bind over here, and Z plus synthesis will be inducible, or beta galactosidase synthesis will become inducible, whereas permease is not being produced, but permease is present over here and the operator is constitutive over here. So permease will be constitutive synthesis. So the answer here is option 3, that is inducible for beta galactosidase and constitutive for lac permease. Then next one. During DNA replication events at the replication fork require different types of enzymes having specialized functions except. So one of the enzyme is not required for DNA replication out of the given options. So that is DNA glycosidase, because it is involved in repair mechanisms. Answer is option 4. Then which of the following names is appropriate for the sequence GANNAUG 3' in the mammalian mRNA? It is called as Cossack sequence. Answer is option 2. Then next one. The specificity of tRNA recognition by amino acid tRNA synthetase that is intrinsic to the tRNA molecule lies on. So it lies on the anticodon loop. Answer is option 3. The effect of nonsense mutation could be nullified by reversion as well as suppression. Which of the following processes will help to distinguish between the two kind of revertants? So what kind of process will distinguish between reversion and suppression? That is recombination. Answer is option 4. Then coming to part C question. Mutants of LAC-Y, that is Y minus, gene of the E. coli do not synthesize lactose permease protein. The following statement refer to the behavior of the LAC-Y minus mutants under different experimental conditions. Which combination of the above statement is correct? So A. No synthesis of beta galactosidase when Y minus cells are induced with lactose. Of course, that is correct, because lactose permease is not being produced. So lactose will not be able to enter. So there won't be any synthesis of beta galactosidase. So A is correct. Synthesis of beta galactosidase when cells are induced with lactose. That cannot be correct. No synthesis of beta galactosidase when cells are induced with IPTG. Now IPTG, it enters the cell independent of the lactose permease. So there will be synthesis of beta galactosidase when cells are induced with IPTG. So D is correct. Then cells induced with IPTG cannot grow in the presence of TONPG. And TONPG is a compound whose uptake is mediated by lactose permease and cleaved by beta galactosidase to release toxic compounds. So once the cell is induced with IPTG, it will be taken up and beta galactosidase will be synthesized. But TONPG, it requires lactose permease for uptake. So it won't be taken up. So cells induced with IPTG, they will grow in the presence of TONPG. So F is correct, E is wrong. So the option here is A, D and F, option 1. Then next question. The semi-conservative nature of DNA replication was established by Michelson and Stahl in their classic experiment with bacteria. They grew bacteria in N15NH4Cl containing medium, washed and then incubated in fresh medium with N14 containing compounds and allowed to grow for 3 generations. So what they did here was, first they grew the bacteria in N15 medium, then they grew it in the N14 medium for 3 generations. Then after that they did cesium chloride density gradient centrifugation of isolated DNA, established the nature of semi-conservative DNA replication. The pictorial representation below shows the position of differentially labeled DNA in cesium chloride density gradient. Had the DNA replication been conservative, what would have been the pattern? So here they have asked for not for semi-conservative replication pattern but for conservative mode of DNA replication pattern. So what would have been the pattern if it was conservative? So first it was grown on N15 media, then it was grown for 3 generations in N14 media. So as per the conservative model, growing on N14 medium for 3 generations, band intensity will be more over there, whereas N15 will have less band intensity. So your answer here is option 3. Coming to the next question. HeLa cell extract was used to study transcription of gene X having 6 introns. RNA polymerase II complex containing all associated proteins was isolated from actively transcribing system and subjected to proteome analysis. Results showed the presence of both splicing and capping enzymes in the complex. When transcription elongation was inhibited by flavopyridol, polymerase complex contained only capping enzymes. So no splicing enzymes were present when transcription elongation was when the elongation was inhibited by flavopyridol. When phosphorylation of the CTD domain of polymerase II was inhibited by a kinase inhibitor, the complex contained neither capping nor splicing enzymes. From these results, following conclusions were made. Identify the correct set of conclusions. Transcription of gene X is coupled to mRNA capping. That is correct. That is why when RNA polymerase II complex containing all associated protein was subjected to proteome analysis, it showed the presence of both splicing and capping enzymes in the complex. So A is correct. Then transcription elongation is coupled to splicing. That is also correct. That is why when transcription elongation was inhibited by flavopyridol, only capping enzymes were obtained, but splicing enzymes were not obtained. So B is also correct. Phosphorylation of CTD is required for the recruitment of capping and splicing enzymes. C is also correct. Both capping and splicing of mRNAs occur simultaneously. That is not correct. If it had occurred simultaneously, then flavopyridol would have inhibited it. But flavopyridol inhibited only splicing enzymes. So the answer here is option 1, A, B and C. Next question. In bacteria, N-formyl methionine is the first amino acid to be incorporated into a polypeptide chain. So what would one think is that all the bacterial proteins have formyl group at their amino terminus and the first amino acid is always a methionine. But that is not the case. So they have given some possible reasons and they have asked to find out the correct answer for the following answers from the following reasons. Deformylase removes the formyl group only during or after the synthesis of the polypeptides. That is correct. Aminopeptidase removes only the amino terminal methionine. Sorry. Aminopeptidase removes the amino terminal methionine as well as 1 or 2 additional amino acids. So C is also correct. So answer here is option 3, that is A and next question. In E. coli, RECA gene is involved in recombination as well as repair. And DNA-B gene is involved in unwinding DNA double strands during replication. It is a helicase, DNA-B. So which of the following statements is or are correct about RECA and DNA-B? Mutation in E. coli RECA gene is lethal. That is not correct. It cannot be lethal. E. coli with mutated DNA-B does not survive. Yes, that is correct because it is a helicase and it is involved in unwinding of the DNA strands during replication. DNA-B after uncoiling DNA double strands prevents further re-annealing at the separated strands. It does not prevent, so C is not correct. RECA gene is involved in SOS response and helps in DNA repair. That is also correct. So the answer here is B and D. The challenges faced by amino acid tRNA synthetases in selecting the correct amino acid is more daunting than its recognition of the appropriate tRNA. In case of amino acids with similar structures like valine and isoleucine, this challenge is met by the enzyme possibly through its catalytic and editing pocket. So A and B.